Hello, my name is Renzo Huber, and today I want to talk about mapping the layer-specific cortical activity with high-resolution fMRI at 7 Tesla. And first of all, I want to thank Alan Wagener for inviting me. It's a great pleasure and honor to be part of this 7 Tesla seminar, specifically in these early days of the seminar. So Alan suggested this seminar format of this pre-recorded video, and then for the live event on September 30th to really focus on the interactive discussion. And I really like this idea and want to take full advantage of it, in the sense that this kind of pre-recording allows me to put different angles on the different sections of this seminar presentation, as well as recording across multiple days, which will make it easier for me. I think I got into touch with Alan first in March and learned to know him when he asked me to update the 7 Tesla map. So I have this blog, layerfMRI.com, which I use, among other things, to keep track of the field of layer-dependent fMRI, and including this map of the ultra-high field scanner sites for human imaging. And Alan asked me to include your new head scanner 7T from GE. So congrats on getting this very nice scanner. I'm particularly excited about your interest to apply this for this scanner for fMRI studies. So most of the 7 Teslas out there are body systems, so they're not really optimized for high resolution EPI or cortical layer imaging for that matter, right? So your scanner can really have a huge impact on the field because it's kind of more optimized for layer imaging. And until now, the field has been really dominated from Europe, which is not really presented by the numbers of scanners. So while there are a lot of scanners in Asia, for example, and in Japan alone there are about 6, I think, they're not really represented in the high-resolution fMRI field just yet. So this means that I'm really excited about your interest and the interest of your center to use this scanner for fMRI, because fMRI is important, right? So already at conventional field strength of 1.5 Tesla and 3 Tesla, fMRI has helped to better understand the brain, how it works, on so many different angles. And until now, a very successful viewpoint to understand the brain with neuroimaging is to look at it at the spatial scale, where we have all these different brain areas that have their unique abilities. Like the visual cortex represents visual perception. We use the sensory motor cortex for moving the body. Listening is done in the auditory cortex, and so on and so forth. And for functional MRI methods developers like myself, it's really quite exciting to help advancing the tools to non-invasively capture the brain activation of these large areas in real time, as you can see here. However, as exciting as it is to see the brain in action like that, it's not really telling us so much about how the brain works. It's somewhat comparable as trying to understand how the engine of a truck works by looking at a truck with an infrared camera. You roughly see which areas seem to be involved and consume a lot of energy, like that the front is used for driving, but we don't really have a deterministic view of the underlying processes. In order to get there, we need imaging tools across spatial scales to also see these kind of small elements of the circuitry. Namely, within the gray matter in the submillimeter regime, we are starting to approach the level of these the mechanics of the smallest neural building blocks, the cortical layers. And these neural layers have very different neurons that do different things and follow predefined information processing algorithms, as shown here. And conventional neuroimaging cannot really distinguish those and needs to lump them all together into these big voxels or pixels. Only high-resolution layer-dependent fMRI has the promise and hope to resolve these different processes. And since these layers have very specific functional connections, any kind of high-resolution activation information will tell the researcher not only if a given brain area is involved in a certain task, but also what this activity means, like where this activity is coming from, how it's following the individual processing steps, how it's modulated and ultimately sent to other areas for further processing with respective feedback, and so on and so forth. So with the right neuroimaging tools across spatial scales, the researcher will know which brain areas are connected to each other, which of those connections are functionally stronger than others, and most importantly, the causality, the directionality of these connections will be known. And previous published proof of principle studies of layer fMRI could actually show this directional connectivity, something that was kind of reserved until now into the realm of invasive preclinical research. So at conventional resolutions, when you ask a person to move their fingers, you do see these heat map-like activation patterns in the sensory motor system here, which is cool to see, but it doesn't really tell us what the brain is becoming active for. Similarly, when you ask a person to use their imagination and just think about moving their hand, the same areas are becoming activated, and the researcher didn't really learn a lot, right? And it might be quite puzzling why we have the identical brain pattern and still it results in so different behavior. However, when one focuses all the efforts onto this tiny part of the brain here, not even covering 1% of the cortex, it is possible to increase the resolution further and further and further until you start to see these stripy patterns of the network logistics, referring to the different cortical layers. So here these pixels forming a line refer to neurons that receive a lot of cortical motor planning input, whereas these pixels here forming a line refer to the corticospinal output to trigger the muscles to move. And with this high resolution, now we can finally see how this is not the case for imaginary tapping, where we don't have this output loop involved because the muscles are not engaged, right? So giving the neuroscientists the chance to see these kind of stripy patterns is very valuable because it tells them not only if there's activity or not, but also it can resolve the individual processing stages. Or at least that's the hope of layer fMRI imaging tools. To get there, however, there are a lot of technical challenges that need to be overcome, which I was asked to talk about today. And I guess from the methodological point of view, one of the biggest challenges still is sensitivity, explained here on this back of the envelope estimation, where you can see that, as you might know, the signal strength of MRI is directly proportional to the amount of magnetization available in the sample, aka the voxel. So going from a conventional 3 or 2 millimeter fMRI voxel to the upper limit of layer fMRI voxels of 0.75 millimeter resolutions is a huge reduction in volume, a factor of 64. And there is no question that we need to make compromises when we work with one of the 64th part of the signal. So we cannot simply go to 50 Tesla scanner or average 4,000 times as long. On top of that, even with these high resolutions, it still doesn't mean that we have the localization specificity. In fact, conventional fMRI gradient echo bolt methods do not really resolve these stripes. Even when we go to higher and higher and higher resolutions, even at 0.7 millimeter, we still end up with these fuzzy blobs. And now we have this blob at high resolution, but it's still a fuzzy blob. And the reason for this is that these gradient echo bolt conventional fMRI methods are mostly sensitive to oxygenation changes in the large vessels that are not as nicely laminar aligned like the neurons are. And this is well known, and the whole field has been looking for techniques to mitigate these large vein contaminations. And there are quite a few technologies out there already. My personal favorite to mitigate these large vein effects is during the data acquisition by using MRI contrasts that are most sensitive to the blood volume changes of the small vessels that are closely located to the laminar neural structures, finally giving us these stripy patterns, even though it comes along with a higher noise level too. And one very effective way to deal with large noise levels is to go to higher and higher field strength, to bigger and bigger scanners. And here in Maastricht, we have 3 scanners available. So I would like to give you a very quick tour and talk about the experiences that I got from using different scanners to get to layers. So here we have one of the very few 9.4 Tesla scanners that work in the world, which is a very big machine dedicated to go to very, very small voxels, specifically for layer fMRI. While the 9.4 Tesla gives great data, it's not so easy to use. So here we have a classical 70 scanner, which is way more user-friendly and also gives good data. However, it's still expensive and not so accessible. So here we have one of our 3 Tesla scanners, which is not really optimized for layer-dependent fMRI. However, it's way cheaper and more user-friendly. So comparing all these different scanners, I believe layer fMRI is doable across all of them. Of course, different scanners have different shortcomings, but they can be accounted for to some degree. So for example, at 3 Tesla, obviously we are more SNR limited. We need to scan longer because we don't have the signal strength as for higher field strength. However, if we push it too much, for example to 9.4 Tesla, then we are also dealing with challenges of working with these kind of prototype scanners that also need a lot of attention from engineering groups, for example. So I believe I can summarize the different field strengths with this metaphor over here, where 3 Tesla is the most cost-efficient way of doing things. It's cheaper, and you can have a lot of throughput. A lot of people are being scanned per day. 7 Tesla is more expensive, but it's also still kind of industrially produced, or for example, the Siemens scanners for that matter, in the sense that they are out there for decades already. They are really over-engineered by that point, so when you push a button, you get layers out of it almost always. And this is not yet true for example, the 9.4 Tesla that we have from Siemens, where you can break records. Yes, you can really get the best performance, better than the 7 Tesla. However, you only get 1 or 2 of these successful experiments out of it every now and then, and you need to have kind of the whole support of an engineering group and really to make it work, to have these record-breaking experiments. So I know that you have experience also in the betweens here. For example, you had a 4 Tesla scanner, right? And I'm not really sure where I would place your head GE scanner, so maybe somewhere between the 70 here and the 9.4T, where you still benefit from all the 7 Tesla literature out there, but still it's some kind of a prototype still. They're not like several hundreds out there. So for example, for Siemens, the 7 Tesla scanners, there are almost 100 out there right now, and about half of them are using these blood volume sensitive layer fMRI methods that I just showed you, with quite promising results for that matter. So across different brain areas and different sites and different applications and cohorts, it gets relatively stable signals. And I'm particularly excited about these first experiments of layer-dependent fMRI in patients at NIH, which I will talk about in a few minutes a bit more. So if we go to 3 Tesla, you don't have as much signal. However, anything you do will have a way bigger impact onto a way larger society. So there are not only a few hundred 3 Teslas out there, but there are several thousands, right? So if you can make layer-dependent fMRI possible, you really affect neuroscience practice and even clinical neuroscience for that matter. So I'm currently working quite extensively to make this possible, and I think a few protocols are out there that seem to be quite consistently stable to produce layer-dependent fMRI results. Here, for example, for sensory motor systems, where you start to identify these stripy patterns in the primary motor cortex, or also in the visual cortex, here for this protocol, and then more in the exploratory phase are these kind of whole brain protocols here to look at movie watching tasks, where you can maybe start to identify how the connectivity patterns follow the cortical ribbon. Now, if we go to 9.4 Tesla, we have way more signal available. However, there are other challenges, and I think for this blood volume sensitive method that I'm interested in, this so-called VASO sequence, we are particularly challenged by radio frequency field inhomogeneities. So for example, if we are interested in a field like there, it could happen that right there the field is just way smaller than in another brain area. And for example, if we want to do whole brain protocols, we really need to optimize the P1 shim, like the radio frequency transmit shims, in a different manner. So in VASO, in the blood volume sensitive method that I care about, we kind of only got it to work by dynamically changing different parallel transmit modes. So here between the inversion and excitation pulses in this inversion recovery sequence that resembles the VASO sequence, we basically flip back and forth between different shim modes for excitation and inversion pulses. And this allows us to minimize artifacts such as these kind of bright voxels over here that represent inflow of fresh blood into the primary motor areas here, and only with these dynamic shims we can get rid of them. But overall, from 9.4 Tesla up to a 3 Tesla, you can apply layer-dependent fMRI. No matter which field strength you're using, I think if you want to go to these very high spatial resolutions, then you also need to make compromises with some other sequence parameters. For example, usually you need to somewhere move in this pentagonal parameter space. So when you want to have very high resolution, you usually need to make compromises in sensitivity or specificity, which we partly talked about with respect to gradient echo bold and this blood volume sensitive method, but also with respect to coverage or temporal resolutions. As soon as you push the coverage, then you cannot easily get towards these high resolutions and vice versa. And I think actually that the coverage was until a few years ago the most limiting factor to really make high resolution fMRI a usable tool for standard neuroscience, right? So I would like to show you a few of the approaches that we've been working on to get towards whole brain coverage with high resolutions. So until now, most of the layer-dependent fMRI studies really focused on specific brain areas, simply because it was efficient. In the first study that I showed you in this seminar presentation about the finger tapping and the imaginary tapping, we particularly focused on this tiny part of the motor cortex. So you might see stripy patterns here referring to the different layers in the motor cortex, but the coverage is tiny. It's not even 1% of the brain. So a lot of effort has been invested into making this entire slice protocol. But then still you don't have whole brain, right? You need to have multiple of those and stack them linearly on top of each other to end up with a whole brain coverage. And this is where we can benefit from redundancies across the different radio frequency channels in the multi-channel coils with accelerated imaging and also advanced sampling approaches. So here, for example, I'm showing you a rather common 3x2 undersampling pattern, so grappa 6 altogether, and you can achieve this in multiple ways. And here it was surprising to me, at least the pattern, the trajectory matters a lot. So for example, you can achieve this kind of undersampling pattern where I'm plotting you the sampled k-space points in solid squares as opposed to the skipped k-space points in white squares with multiple different acceleration approaches. So for example, in your 3D k-space, you can do a 2x3 undersampling pattern and then do a chi-p shift into the Z direction, right, with the conventional blipped chi-p. However, you can also achieve the very same undersampling pattern by not going through k-space in this kind of down, up, 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 down pattern, but instead using a 6-fold acceleration only in ky, but then use different pre-phaser for the different shots in your excitation pulses to end up with the same undersampling pattern. However, the trajectory is very different. And you can achieve this even the same trajectory and same undersampling patterns by using segmented approaches. For example, here a 3-fold segmented approach which then has set-wise pre-phaser chi-p, which ends up with the same trajectory. And here you can see that the same undersampling pattern, so the same acceleration, can result in highly different image quality. So here we benefit a lot by using shot-wise acquisitions of 3D k-space where we orthogonalize the low bandwidth direction and keep it as short as possible from the shot-to-shot B0 and phase inconsistencies in the k-z direction, right? So with this we can benefit a lot and get quite nice, almost whole brain coverage data. So here I'm showing you a movie watching experiment where I show you the conventional networks. Here, for example, the frontoparietal network. I think this one, now I missed it and my computer is a bit slow here. I think this was the characteristic RIs of the default mode network. It comes up again now. So we have the sidewise RIs and this one over here. Here we have the frontoparietal network, or also the interparietal cortex, which can be better seen here in the stills, not the movies, where you see that there are these very specific layer patterns visible. For example, in the default mode network here we have this kind of one line following the cortical ribbon, whereas for example here in the interparental sulcus, or also here we have these kind of double stripe patterns. So there are very clear layer features of signal variations, local signal variations within the cortex popping up. So this convinces me that there is some signal that we can harvest in the future, and we are just starting to ask questions on what these signals means. Do they refer to neural activity? Do they refer to the vascular architecture there? But it definitely tells me that it opens up a whole new field to look at connectomics, right? Because in conventional resting state connectivity or natural like naturalistic task connectivity, you usually do an fMRI and then you chunk up the brain in all these different brain areas and you look at the correlation of each brain area to each other brain area. So you have this kind of connector matrix and then draw conclusions based on that. Now with layer-dependent fMRI, we have a whole new dimension onto that. So we not only see which area is connected to which other area, but also which area is the receiving end, or so we get some new ideas about the directional connectivity there. So here I just want to show you a few example networks. Here, for example, the default mode network, or the visual network, or the motor network, or also the frontoparietal network, where you can see what kind of new information we can get from these kind of layer-dependent connectivity measures. So in the default mode network, for example, you can maybe see that PCC ROI kind of stands out in the sense that it's the only area that's kind of middle layer dominated. In the Velleman-Van Essen canonical microcircuit, the middle layer is the feed-forward input layer. So you can see down here that in the PCC we have this middle layer peak, whereas the PFC or the IPC are kind of having this additional superficial peak over there. So they're also receiving a lot of feedback input. So it seems that the IPC and PFC are kind of passively being dragged along by the actual hub of the default mode network, the PCC, which is driving the network. The other ones are just being dragged along. We can try to kind of validate these rationales of using layer-dependent fMRI for directional connectivity with networks where we already know the hierarchical layer structure. Here, for example, in the visual system, where we know that the primary visual cortex V1 sends feed-forward input to V5 in the kind of middle, deeper layer 4, the input layer into V5 from hierarchically deeper brain areas, whereas the other way around, V5 sends feedback to V1 almost exclusively to the superficial and or deeper layers. And this is even clearer in the motor cortex, and the motor cortex is particularly nice for validating layer fMRI in the sense that it's 4 millimeters thick. It's like almost double the size compared to other cortical areas. So in the primary motor cortex, we do see that all the feedback input from the sensory area, so any cortical input for that matter, almost exclusively comes into the superficial layers. The deeper output layers that trigger the muscles to move, they don't really care much about the sensory input so much. And then also we can try to even approach other networks where we don't really have clear hypotheses. Here, for example, the parietofrontal areas, where you might be able to see that in the parietal sulcus we have these clear distinctions of kind of superficial and deeper layers. They almost behave like different cortices altogether, and sometimes this is even directly visible with different ICA components. So now that we are kind of convinced of the usability of layer-dependent fMRI across larger field of use for connectivity, I think I want to generate a kind of atlas of a whole brain layer-dependent connectome. And this is where a study where I'm working currently on, particularly with my student Anna Müller, and the study is now being continued by Kenju, who is from Japan actually, and is working with me on this for the next 9 months. Kenju is originally from Yuichi Miwaki's lab in Tokyo. So in this study we are acquiring a whole brain layer-dependent fMRI protocol during a movie watching task, and we average the heck out of it. So there are 5 movies per session and at least 5 sessions per participant. The first data set is already uploaded and the new one is already collected and being collected at this very moment, and then hopefully uploaded until end of the year. So we encountered quite a few challenges in doing this. For example, to do these kind of very high accurately like alignments for layer-dependent fMRI in a space where we are limited by low bandwidth and distortions, we need to do kind of non-linear motion correction, because suddenly the brain does not really behave rigidly anymore across many days and runs. We learned how to do layer fMRI directly in the distorted EPI voxel space and have decent TSNR, or at least double-digit TSNR across almost all cortical areas. And this allows us during movie watching to do all kind of unconventional connectivity analysis. So here we use the same movie that is used for the 7 Tesla HCP study. This means that we almost already know what to expect on a low resolution scale. So from HCP, low resolution fMRI, we can take the non-sibling data from 93 participants, and while watching these same movies, and extract signal time courses across the entire brain, and the entire brain is involved somehow during those movies, and then can use them as regressors to kind of extract brain areas from any area that we want. So here, for example, some new areas that work quite hard until now to be accessible with layer-dependent fMRI are kind of the auditory areas, speech, or here just the loudness of the movie. But also the default mode network has been particularly elusive, and it comes out pretty nicely in these movie data. As soon as we move from these primary cortical areas to more cognitive areas, however, we have this challenge that we need to kind of calibrate the cortical depth that we get from layer-dependent fMRI with kind of the cytoarchitectonically defined layers. So for example, according to Brodmann, there are 6 layers, there are other definitions, but we need to kind of get some correspondence in there. And this is where we benefit a lot from kind of ex vivo atlases. Here, for example, these Ding atlas where they did ex vivo MRI, then sliced up the brain and looked at histology. So we approximately know a correlation for each different brain area between histology and MRI, and then can do the same kind of high resolution MRI in vivo and try to find these certain landmarks. Here, for example, let's say we have this elbow structure, so we know that this elbow is probably happening in layer 2-3. We know this from ex vivo. This means that this bump here that we do see in layer-dependent fMRI is probably coming from layer 5-6 or anything below layer 4. And you can scroll through these different brain areas and see how variable it is, the blood volume responses in these connectivity analysis. And there, because each different brain area does have different thickness of the layers, we can now then calibrate which depth refers to which cortical thickness. So now that I talked about all the acquisition challenges and the hardwares and the scanners, I would like to shift gears a bit and talk more about analysis, the pre-processing. And this is where I benefited a lot from Farouk Gülban. He's a colleague here in Maastricht, and together with him we developed the layer fMRI analysis software package called LANY, that is particularly designed in order to make layer-dependent fMRI easier for more general neuroscientific applications. So not only for these like whole brain acquisitions, but also for kind of the standard layer-dependent fMRI as you find it in the field. LANY stands for Layers and Nifty. It's written in C++ and easily applicable in Linux, macOS, and Windows. Installation instructions, example pipelines, as well as tutorial videos are listed on GitHub. The reason why we've developed LANY is coming from the challenge in layer-dependent fMRI that we have very high quality requirements for the analysis, still while working with these messy, non-standardized data. So for example, the high resolution in layer-dependent fMRI is often only achievable within acceptable speed by using these small field of views that do not fulfill the common topology requirements of mesh-based layerings, namely having kind of closed surfaces without holes. And without these requirements, smooth surfaces are usually not well defined and we end up with unconnected spots as seen here. So we need tools that allow us to work with such messy data. Another challenge is what I call the beer-wine dilemma. Namely, in the encoding limited regime of submillimeter fMRI acquisition, we are usually limited by participant-specific MRI artifacts so that the acquisition protocols are commonly highly optimized for the goal in a given study, and the data cannot be easily pushed through the pipeline that has been developed for another study. So while conventional fMRI, representing the beer here, has been standardized over decades to give you data that are homogeneously be pushable through standard pipelines, like done by multinational brands of beer corporation, this is not really doable with layer-dependent fMRI representing the wine here. There we have specialized data and specialized study protocols and challenges that need much more interactive flexibility, quality control, especially in these early days of the field. So like wine, you get the best quality by not homogenizing your data to be pushable through Henry Ford-like assembly line production, but rather we need flexibility across like regions and years for wine or the data types that the user is working with. So we need layer-dependent fMRI tools that allow a flexible balance of user interventions on one hand without the danger of biasing your data on the other hand. Furthermore, in layer-dependent fMRI, unconventional non-bold contrasts like VASO or VAPOR are being popular, which consist of these control label images that need to be dynamically sorted without the danger of noise amplification. So we need convenient tools for those too. And last but not least, layer-dependent fMRI data are also particularly challenged by low bandwidth artifacts and the very low SNR, which needs to be addressed somehow during the analysis. So these are all layer fMRI challenges that most software packages have never been built for. So I would like to show you now some of Laney's example programs to address these. I guess one of the most popular features in Laney is the layerification of small field of view data in voxel space. So here, for the sake of validation, tested on a small field of view section of ex vivo visual cortex. And at these very high resolutions, you can see that the equidistance layering kind of underestimates the curvature of the stria genari. Thus, Laney conveniently also spits out the equivolume layering that fits the stria genari quite nicely. Another example of validating Laney's layerification here is the 100 micron whole 3D big brain dataset, again showing that at these ex vivo resolutions, the equivolume layers fit the histological layers quite nicely. Another core program in Laney is the columns generation by means of iteratively dividing the 3D voxel ribbon, allowing the user to unfold and flatten the cortex without interpolation of vertices, but as a direct voxel intensity remapping, which can be very useful to depict topological fMRI data like the somatotopic arrangement in the sensory motor system. So here taking the central sulcus as a two-dimensional voxel grid, which then can be used to flatten it orthogonally across layers and sulcal depth to ultimately end up with a 3D nifty volume of layers and columns completely orthogonalized. Laney also contains deveining tools using model-based approaches to minimize training main effects of gradient echo bold data, using previously published formalisms to convert the superficial bias in gradient echo bold to make it look more like non-bold contrasts. In cases where you can assume that neighboring voxels and neighboring columns have very similar layer activity, layer-specific smoothing without signal leakage across layers can be very helpful to deal with the noise level of individual runs. So now that we have all the acquisition and the hardware that we talked about and all the analysis tools, Laney, we can go to the clinic, as you can see in the background. And here I would like to show you an application study, a clinical application study of layer-dependent fMRI done together with Silvina Horwitz at NIH in US. And I already here present you three particular challenges that we encountered when doing layer-dependent fMRI in the clinical context. And those are additional challenges on top of all the other layer-dependent fMRI challenges. And one of the most obvious challenges is that those patients are not trained participants, right? They move more. So in the motion traces, you can see these spikes, right? When patients are uncomfortable, then they position themselves new and then we have more motion. Also, they are not as kind of motivated to follow the task as much as, for example, that the conventional neuroscience students. And I'm not sure how well this comes across here, but here this is a finger tapping task, and you might be able to see that when they're supposed to move their ring finger, the pinky finger kind of moves along with it. So we have a kind of loss in the task specificity or kind of contrast to noise ratio to distinguish one finger from the other one. And also, we don't have as much time as we would usually have in the protocol setup for each participant to optimize the acquisition. So we just need to take whatever we got, even though the images are usually kind of compromised by artifacts, for example, these kind of interference artifacts. So we need to mitigate those individually. And for the motion, there is not so much you can do, but just manually go back, look at each individual block without motion, and then align it manually before you run the standard motion correction in order to give it kind of a good starting point. In order to deal with the noise level or the task insensitivity, we benefit a lot from the kind of different signal pooling and smoothing tools in Laney across columns and layers depending on the research question. And these artifacts are also significantly reduced when using blood volume sensitive methods because they are inherently normalized. They're always pairs of images, so they're not as sensitive to the baseline arbitrary MRI signal. And with all these different tweaks, we get quite consistent results across patients. So here there are data from 4 patients, both on the affected and unaffected side. And those are patients of focal hand dystonia. So this is a quite common neurological disease where patients cannot move their hand properly for certain tasks only. So for example, for writing or some of them for playing a musical instrument. And we have 4 patients and 4 healthy controls. And for the 4 healthy controls, we didn't have as many issues, of course. And so that's why I'm showing only the patients here. So it does work. And in order to make you more familiar with the kind of analysis that I want to show you and the data types, I would like to show you how in LANI we use kind of the tools LN multilaterate and LN patch flatten to kind of look at folded cortex in a kind of a flattened view. So imagine we have any kind of a 3D voxel like this one. And with the new tools included in LANI by Farouk, you can select any kind of seed voxel, and then it's using a kind of flooding or growing approach to span cortical distances. And as soon as you have cortical distances, you can have regional relationships and basically span a cortical grid shown here. So within the voxel, voxel space, you see the kind of folded structure, and then you can flatten it here for the columns or then also for the layers. And all of this is just a direct volume to volume remapping. So there are no surfaces or interpolations involved for that matter. And on the left here, you can see how this kind of topographical analysis in LANI looks in the kind of native distorted EPI space, where we have this kind of orthogonal grid coordinate system and then axial view along that. So in the individual projections, sagittal, coronal, and axial, you don't really see the columns that well, or it looks messy, maybe not so much in the axial view, but in the 3D view, you see this kind of sausage candy pieces stacked together. So in this kind of flattened column by column view, we directly with the naked eye see how the affected hemispheres in those patients are showing us different topological representations of the different fingers as opposed to the affected side. So affected side is right, a healthy control side of the same participant here is on the left. And you can see in the healthy side we have this somatotopic arrangement, meaning that the index finger is arranged next to the middle finger, next to the ring finger, next to the pinky finger. Whereas in the affected hemisphere, it's all completely messed up, right? It's not for the lack of sensitivity, right? We have a lot of activity there for tapping-induced activity. However, there is no clear discernible kind of structure. There's a lot of overlap, and we hypothesized that this is coming from a lack of surround inhibition of the individual finger representations. And you can maybe even glimpse at which layers these different surround inhibitions are coming from. So here I'm showing you the layer profiles in the motor cortex for the affected and unaffected hemispheres. And you can see that for the patients in the affected hemisphere, we have this kind of lack of inhibition in the superficial cortical input layers as opposed to the unaffected healthy controls in green here as opposed to the affected side in blue, where the blue side is really dominated from the cortical input superficial layers. And this is not as clear, but also still visible in the conventional gradient echo bold data, not only the blood volume sensitive RASO. So these data make me very excited about the usability of layer-dependent fMRI and its applications not only in neuroscience, but also in clinical neuroscience, which will hopefully really increase the impact of layer-dependent fMRI to a wider field. To conclude, transformative brain imaging technologies bridge many different spatial scales, all the way from the macroscopic side of behavior, manifestations of mental disorders and the brain areas, to the microscopic side where we have synapses or the cells. And each of these different spatial scales have their transformative imaging techniques. The missing link of methods to obtain a more comprehensive understanding of the brain is the mesoscopic part of a few hundred micrometers, where we have these circuits of the computational building blocks, the layers. And this is exactly where the value of these high-resolution layer fMRI non-invasive imaging tools come in. And with this, I thank all my colleagues and I thank you for your attention.